Welcome everybody to this second webinar in Polyphenol Research. These three webinars are organized by the Groupe Polyphénols on various aspects of polyphenol sciences. So my name is Denis Barron. I work for Nestlé Research and I am the president of the Groupe Polyphénols. After a first session on polyphenols in natural soil systems, today our topic is the characterization of polyphenols in complex mixtures. Actually, why did we choose such a topic? Mainly this is due to the fact that we are not dealing with single chemical entities like drugs, for example, could be, but with natural products that naturally occur as complex mixtures and exerts their biological activity through combined effects involving multiple targets. So, of course, this is a very complex situation, and how can we, as scientists, deal with it? So today I have the pleasure to welcome two speakers who will share with us their experience in this topic. So I think that we have now two slides to present the speaker. Thanks a lot. Our first speaker is Professor Jean-Luc Wolfender. Professor Jean-Luc Wolfender is a natural product chemist who obtained his PhD in pharmacognosy from University of Lausanne in 1993 under the supervision of Professor Kurt Ossetman. Then he moved to University of California at San Francisco for a postdoc in the group of Professor Burlingame. He is currently full professor at the School of Pharmaceutical Sciences of the University of Geneva, and he is also the vice dean of the Faculty of Science of the university. So actually, really, Professor Wolfender has been a pioneer during the 90s in the introduction of LC-MS and LC-NMR techniques for the chemical characterization of crude plant extracts. He specialized in the identification of novel bioactives using a combination of comprehensive phytochemical profiling and systems biology approaches. Our second speaker is Dr. Delphine Winstel. Yes, exactly. After a master's degree in phytochemistry and cosmetics at the University of Corte in Corsica, Dr. Winstel obtained her PhD in enology in 2019 from University of Bordeaux under the supervision of Professor Axel Marshall. Since 2020, Dr. Winstel is a postdoctoral researcher in enology at the Institute of Wine and Wine Sciences of Bordeaux. So the work of Dr. Winstel focuses on taste active compounds of oak wood that are involved in the taste balance of wines and spirits. Now, in terms of practical organization, our two speakers will first deliver their presentation, and we shall collect all the questions at the end of the session. So you can ask questions through the chat. The session will officially close at 3 p.m. GMT. Nevertheless, in case we have many questions that are still not answered, our speakers have accepted to stay connected a little bit longer to answer them. So if you are still available, you can still stay connected after the official closure of the webinar. So now I have the pleasure to give the floor to Professor Jean-Luc Wolfender, who will discuss the efficient composition assessment of phenolic compounds in plant extracts. So, Jean-Luc, please.
Perfect. Thank you very much, Denis. I hope that you can see my slides. So thank you very much for the invitation. And indeed, today I would like to quickly summarize our metabolite profiling strategies on different examples of natural product, and I will also try to deal with a few polyphenols. We are mainly working in in pharmacy, and our aim is more to isolate efficiently bioactive compounds from natural sources based on this reductionist approach, which is bioactivity guided fractionation, where we start from extract and we end up with pure bioactive natural product. We can also today, with new methodologies, to have a more holistic approach where we try to get the metabolite profile of the extract at the beginning of the process And through annotation, we can guide also the annotation on selected product in a more efficient way, and that's what I'm going to try to show you today. So we will mainly speak about metabolite profiling, and I will try to show you our strategy to do metabolite identification based on these profiles. You probably are aware that one of the gold methods now to do profiling of plant extract is mainly LC-MS. And we use especially UHPLC MS with high resolution to try to decipher the composition rapidly. And we combine this with a technique called molecular network. And I'm going to introduce this technique to you to show you a little bit how we use it. So the whole challenge is, of course, to annotate correctly the structure of all these different metabolites, mainly polyphenols in your case. And the challenge is the fact that we don't have access to generic databases of all natural products. So we have to find out either a way to identify them by targeted isolation or interpretation of spectra. But mainly what we are developing at the moment is databases containing in silico spectra for spectral match. And we try to putatively give good annotation of most of the constituents. If you look at an LC-MS profile, this looks like that. So you have this scale here, which represents the retention time, the other scale, which represents the mass scale, and this type of maps are extremely sensitive, and all the peaks you see are correlated to compounds. And the methods today are extremely powerful with LC-MS, and you can annotate the main constituents that you see here, but also the minor ones. And depending on your platform, you will be able to retrieve the high resolution mass spectrometry spectra of each of these constituents, which allows you to find out the molecular formula. And at the same time, you can trigger an MS-MS experiment to have a fragmentation spectra, ideally on all of them. So you will see that this is generating hundreds of spectra, even thousands per extracts. And now you have to find a way to data mine all these extracts. So we will look at these spectra more as fingerprints or barcodes. So in a typical extract that is profiled here with extremely high resolution in about 80 minutes, this is the type of ion map that you can get with LC-MS, having in mind that each of these dots correspond to an MS feature, and most of them just correspond to single constituents. So you see that this is extremely complex on any plant extract. This is an Arabidopsis sample, for example. And for all of these dots, on the MS1 side, you will be able to retrieve the molecular formula, and on most of them, you will get also the MS-MS spectra. So this is the type of data that we are dealing with. So now, in order to try to annotate all these compounds, to do what we call a deep metabolome analysis, first we will use the different algorithms to extract the molecular formula and use different filtering methods. I will not go into details, but today you can annotate most of the molecular formula rather easily. We will combine this information with taxonomical information, which is important in natural product chemistry, in order to be able to be more confident in the annotation of these compounds. Of course, this is still an annotation based on taxonomy and molecular formula, and this is a first idea of what could be the constituent. You have to go in more depth, and for this we use the MS-MS spectra of all these constituents, and we process this data in the form of molecular network analysis, which allows us to group all the different constituents based on their structural similarity, and then we annotate every single MS-MS spectra. I will try to show you quickly how we could do this. And finally, whenever it's needed, we can perform targeted isolation of the compound of interest to go to sensitive micro-NMR method for full de novo identification. So if we go back now to this chemical profile, I would like to show you how we can build this molecular network of all the different constituents. So the principle of molecular network is the fact that you can look at all the MS-MS spectra of all the different constituents that you have in an extract, And through an algorithm which will vectorize the data, you can compare the similarity of the spectra. If two spectra are similar, they will generate dots in the molecular network which are close together. If they are very dissimilar, you will get another cluster. So this is a spectral similarity network, but this, as you know, the MSMS spectra of a molecule is closely linked to the structure. So that means that if you cluster together spectra, you cluster together as well structures. So if you apply this to an extract, you will see cluster of ions which goes together, and normally the annotation of all of these compounds will be also rather similar. So that means that when doing this, you can start to cluster together analogs of the same structure. And if you do this on one extract, typically you will have different cluster of spectra. And if you have a cluster, this means that you have related analogs, related flavonoids, related stilbenes, etc. So this is quite useful when you look at the composition of an extract. I would like just to show you how we could do this practically, not only on one extract, but on a collection of extracts. In this case, for example, we work on about 300 U4BAC extracts. We were interested for their diterpene contents. And what you see here is not a science fiction movie. This is actually the calculation of the MSMS correlation of all the different constituents which were detected in all the 300 extracts. So this is about 1 million MSMS spectra. And what you see on the screen is the building up of the network of all the different constituents detected. So at the end of this calculation, you end up with a table like that, where you will have a lot of different clusters. Actually, each of these clusters will contain analogs of the same structural type. And you see in this case, we were having about 1 million spectra and about 2,000 clusters. So if I now go on one of them to show you the type of information that we have, this is typically how it looks like. And every dot will correspond to a mass spectrometry feature. So it's related to a single compound. And in this case, for example, we could identify two of them because we were having the standard. This was a collaboration with Marc Litodon at the ICSN. We were having the standards. So these two dots were fully identified. Since they are all together in a cluster, this means that what we have in the cluster is closely related analogs. And if we look at the molecular formula of the other dots and the literature, for example, in this case, we could dereplicate, as you see, quite a lot of these other diterpenes. And for other dots, other features, there was no annotation. This means that probably we have new compounds. Also, if we zoom into one of these dots, the information which is there is the mass of the different constituents. For example, here, this was an ion with a mass 733. And you see different colors of what I will call a camembert. And this means that this ion, for example, in the 300 extract, can be found in 3 different extracts. In this case, this was in the leaf, the wood, and the bark of the same species. But you can also have the same ion in different species, and this is interesting when you start to correlate such information with bioactivity data, for example. So you see there is quite a lot of information from this molecular network. So this is the chemical side. So this is what you have on the top of this slide. But now, since we are doing drug discovery on our side, we would like also to map such massive molecular network on hundreds of extracts on bioactivity mapping. So we will also correlate this with the activity of a given extract. So for this, we have the chance to work on a very large library of extract that we get in collaboration with Pierre Fabre Company. And what you see on the screen is a collection of more than 15,000 plant extracts, which were collected by Bruno David at Pierre Fabre. And all these extracts have been negotiated, so we can work on them, and there is no issue with the Nagoya Protocol by working on these ones, for example, for scientific aspects. So if we build up now a molecular network on not on the 15,000, we have worked at the moment on about 10% of the collection, about 1,600 extract. You see you can build up a huge network, which is very informative. Now, the idea for us would be, since we have annotation on all these constituents, of course, with not always with the right structure, but with an estimation, our idea is to convert this library of extract in a chemical space of individual natural products. And you probably know that chemical space is used in medicinal chemistry in order to navigate and use this for example, in drug discovery program to look for specific ligands. So then, if we have all this putative structure of all the different metabolites in the extract, the idea is to use artificial intelligence to guide the exploration of the chemical space and do targeted isolation of valuable natural products only. So how to deal with this? This is a little bit like in metabolomics. We are looking for needle in a haystack, and you know that it's quite tricky to do this. I have a very nice team of very talented young researchers, mainly developing now a computational tool to investigate such a massive set of data. And the idea is first to do annotation of the compounds, and for this, since there is no real database with real spectra which is available, we use all the compounds which have been published in the Dictionary of Natural Products that maybe you know, and based on the structure, you see here the chemical space of the different products which have been reported. Based on this structure, we could calculate in silico the MSMS fragmentation of each of these published structures. And now we use this library, which is containing about 125,000 MSMS spectra calculated in silico, to map this against the MSMS spectra that we have experimentally. Of course, the match is not very good, but this is an approximation, but we use this plus taxonomy in order to ascertain the structure. So now I would like to show you how we could do this. So in a first step, what we will do is try to assign the structural type to all of these metabolites, and this is now possible with software like Canopus or Natural Product Classifier, which are tools developed by computational chemists, which are freely available and you can use on such type of data. So this will classify your sample in alkaloids, carbohydrate, flavonoids, etc., based on the color. So as you see, you have clusters which are based on structural type. But this is not enough. This is just telling you what type of compound is behind a given LC peak. What we would like to have is more information on the annotation of individual metabolites, and for this, we need taxonomy information. In order to build up the taxonomy knowledge, thanks to two of my, one of my PhD and my postdoc, Pierre-Marie, we built up this year a database called Lotus that every one of you can use, where we try to filter all what is available publicly in the literature by making a link between the taxon and the chemical structure and add a reference which is documenting this. This is very important because this allows us now to link a given structure in the form of an Inchi key to a Latin name and to a given DOI for the publication. So I will show you that this is very helpful because we have now 700,000 reference structure per organism that we can use in order to enhance the annotation. And if you want to try the tool, you can just go on this website to use Lotus. And what I show you here is the chemical space of all the compounds which are found in Lotus. And the different colors correspond to different structure type. And if you zoom into this, you can see, for example, how we could organize the compounds based on chemical similarity in such a nice chemical space. So this is just to show you that we can summarize all this in a chemical space, and then we can explore this chemical space. Now I would like to show you one example of polyphenols we have been working on For example, in a nice collaboration with Thailand, I had a PhD, with Malaysia, PhD at Dinabzon, who was willing to analyze the 7 varieties of a ficus species. So for example, if you hit ficus in lotus, you will get 610 structure related to this genus. And now we can use the annotation and this taxonomical information to improve our annotation. So typically for this ficus species, we can build up a molecular network as it is shown here on the 7 species. And in this case, we get about 1,500 spectra for this different ficus species. And I just zoom here into a few clusters which were obtained for some of the flavonoids found in this species. So now I will show you that for one of the clusters, C11, which is here, how we could now ascertain better the structure based on taxonomy. So first, if we compare all the spectra of these flavonoids to simulated spectra, we could rank them according to the similarity, and the best one is ranked in the first position. If now we use information from Lotus, knowing that we are working on a ficus species and taking into consideration the taxonomical relationship between the sample, we can re-rank the annotation. In this case, for example, one of the spectra which was ranked number 4 for a given structure goes in number 1. So this allows us to be more precise and confident in the annotation. And we could do this, for example, here on this cluster of flavonoids. What you see on the screen was our first annotation, only based on MSMS spectra. And by looking at the annotation and the taxonomy, we could see, for example, that this aglycone was not making sense based on the other aglycone in the cluster and based on the taxonomy, and we could correct the structure. So this is just to show you how we could use taxonomic information plus MSMS to try to be more confident in annotation. So based on this, for example, on this ficus species, first we isolate 24 standard. So this the structure of this standard can be found on the upper part of this slide. So we were very sure about 24 annotation, but now, based on the molecular network analysis and based on the analog relationship, you will see that we can spread the annotation to all minor constituents. So for example, from 24 standard, we could annotate now 130 metabolites of the flavonoid type in this profile. We could have 95 organo-oxygenated compounds, 200 lipids, and 152 fatty acids. So you see, even for the very minor constituent, we could have a very extensive coverage of all compounds. And we could annotate 500 constituents out of the extract. Now the question, of course, is to know what are the main one and what are the minor one, and we are also developing tools that I have no time to show you today on these aspects. Now, if we turn back to the library of Pierre Fabre sample, how can we prioritize samples? So we developed 2 tools which have been published this year. One tool is called Inventa, and this allows us to look at all the molecular network data of these 1,600 spectra, and by this algorithm to highlight all the nodes which could correspond to new natural products. And these nodes will become bigger, and in these, as a proof of concept, for example, we were able to prioritize one sample, which is Pristimera indica, a plant of interest, because this is a Celastraceae, and based on the molecular network analysis, we could see that many ions were corresponding to agarofurans, and based on our prediction, we were quite sure that these were new natural products. They were specifically isolated, and indeed, all of them were new natural products. So you cannot use the same thing to find, for example, new polyphenols if you are interested. Then the last step is to try to correlate this network to bioactivity, because we would like to find bioactive molecules. And for example, we have also developed algorithm which allows us to highlight the cluster which are in connection with the bioactive extract. So for example, we were interested by anti-cancer agent for the Wnt signaling pathway. And I would just like to show you now one example, always on the same network. You have seen this network on the 300U4BAC. And now we screen the same 300 plant extract against Wnt signaling, and 20 of them show bioactivity. So we developed methods which allows us now to color code the different nodes of the different constituents in the network. All the non-active compounds were colored coded in blue, which was the majority of the whole network. And the bioactive one, 20 extracts, were highlighted in red. And if you do this, this is actually adding metadata to your nodes. If you do this, you will see that among the 2,000 cluster, we could highlight 21. So this is really a very efficient way to target the isolation of compounds. And for example, this cluster here, we could replicate this cluster. It was we knew that this was a 4-ball ester derivatives, or as the structural type. We didn't know the exact structure. We knew that it was very different from non-active cluster. And through targeted isolation, now we could prove the isolation, the structure of the compound of interest. In order to do this, what we do is that we go from the chemical profile straight to the semi-preparative HPLC, and we have method which allows us to keep a very high resolution as well to the semi-preparative side. And this is quite nice because then we can target the isolation of a given compound. And the expert for this on our side is Emerson Ferreira-Queiroz, who is associated professor in my group, and you see that he can work at the analytical scale, at the flash scale, or even at the MPLC scale. And we can use the same calculation. Of course, this is working better at the semi-preparative scale to have a quick isolation. So for example, just on this example of U4BAC, we evidence in our collection this nice 4-ball ester derivative, which was a very potent Wnt inhibitor in our assay. And by looking at the structure, we find out that it was very closely related to tigdylanolone, which is another 4-ball ester, which is currently in clinical trial phase 2 against a given type of cancer. You see, so this was a nice proof of concept that we can pick interesting compounds in this data. So we can use the same data now to build up a chemical space, and for this we are using what is called a minimum spanning tree. This allows us to convert the molecular network, which is difficult to read, into a tree where all the molecules will be linked together. And this can be organized as the Lotus database that I showed you before. So for example, this is now the chemical space of Lotus again. And I just show you, for example, by zooming in here, that indeed in part of the tree we have molecules which are very similar. They are just organized in this way. So this is, for example, molecules from CIMA or rubacea. And this is true for every type of data. I have just now to switch to the next slide. Okay. So this is the literature data. And now you have seen that with our methods, we can annotate compounds, and we can use exactly the same format. And what you see here in blue is all the annotated compound that we have on the 1,600 extract of the Pierre Fabre library. And now we can see if there is similarity between this compound and what is known in the literature, and we can make connections. So we can decide to select given analogs, or we can decide to go in another part of the chemical space to do targeted isolation of compound of interest. So this is now work in progress on our side, and the idea behind is indeed to build up a chemical space of all annotated compound, of course with a given accuracy, and be able to target the isolation of valuable natural products only. So this was a rather quick summary with new tools. Most of the tools that I show you are on the web. You can use them. They are all based on LCMS data, and our aim is to now be able to mine all this data to make connection to taxonomy, to make connection with the molecular network, to integrate this data with in silico databases to do the annotation, and finally make correlation with different type of bioassays to predict what could be a metabolite in an extract which is bioactive, and then finally to target the isolation of such compounds. So we hope that this is an interesting method which allows to go more deep in metabolome studies. I could not show you so many polyphenols, but this is very applicable to polyphenols as well. And then target the isolation or the comparison on extract of interest and select interesting pure natural products. So with this, I would like to thank all the people involved in my group. As you see, they are quite active. This is a rather old slide, actually. So Emerson is in charge of the isolation. Pierre-Marie was in charge of most of the computational aspects, for example. And I'm really thankful to all of them for their help in such type of studies. And I just finished with a picture of the building of the School of Pharmacy here in Geneva. I'm speaking from the 6th floor, somewhere here. And we have the chance to have a nice NMR platform with a 600 megahertz system with cryoprobe for final structural identification. And we work on an Orbitra platform for most of the LCMS-based metabolite profiling studies. So I thank you very much for your attention. And thank you very much, Denis, as well.
Thank you very much, Jean-Luc, for this very interesting presentation of those exciting results. I'm sure I hope we'll have a little bit of time to discuss all that a little bit. So now I will give the floor to Dr. Delphine Winstel, who will discuss the complementary approaches to discover new taste active compounds in wines and spirit. So please, Delphine.
So first of all, I would like to thank the Groupe Polyphénols for this invitation. So my presentation today will be about the development that we made in the ISVB for the discovering of new taste active compounds in wine and spirits. So this project took place under the supervision of Professor Axel Marshall in the University of Bordeaux. So for several years, the chemistry of taste has aroused high interest both from academics and industrials. And new taste-active compounds have been identified in various plants, exhibiting mainly sweetness, but also bitterness. Here I put some examples for each taste. You can see that these molecules belong to diverse chemical families, and their sensory properties are strongly affected by slight structural modification. As a consequence, the investigation of natural taste-active products in a given matrix often appears as a major challenge for chemists. Such studies are particularly interesting for enology, since they allow a better understanding of wines and spirits taste. The pleasure felt during a wine tasting depends on many factors, among which its sensory quality plays a special role. As you may know, wine quality is not based only on aromatic typicality, but also on gustatory balance. These balances rely on sweet and sour taste of white wines and sweet, sour, and bitter taste of red wines, according to the famous enologist Émile Pénaud. So, but where does the taste of wine come from, and what is the origin of sweet and bitter compounds in wine? First, compound could come from the grapes, with the effect of grape varieties, but also terroir effect. They could come from the fermentation, for example, with some compounds that could be released either by the metabolism of yeast and bacteria or their degradation in wine, but also compounds that could be released by the wood into the wine during the barrel aging. Of course, the taste of wine can be influenced by the aging of bottles. In the laboratory, we worked on the first 3 matrices, but today I will only present you the results obtained in wood. So barrel aging is a crucial step, which is involved in the development of most great wines, red or white, and spirits can be also aged in barrel aging. From a gustatory point of view, barrel aging also influences the taste of the wine by increasing its sweetness, but also its bitterness. However, there is a lack of knowledge about the molecular explanation of this sensory phenomena. But when we talk about the bitterness of the wine, we often mention tannins, a major class of oak wood. However, studies on elagid tannin have mainly focused on chemical characterization and not on their taste. So what about their sensory contribution? Results show that the detection threshold of the main elagid tannins were significantly higher than their concentration in wines, suggesting their low influence on wine bitterness. And so far, no correlation between bitterness and oak tannins has been clearly established. This compound cannot explain the gustatory importance of oak aging, so a large number of non-volatile molecules released by the wood into the wine remain to be identified, as well as their sensory contribution. So to isolate this type of molecules, an inductive approach with a fractionation protocol guided by gustatometry has been implemented in our laboratory for over 10 years. So this inductive approach involves an extraction step, followed by the development of a fractionation protocol. Then this extract was first partitioned by liquid-liquid extraction with solvent of increasing polarity, and extracts were obtained. These pre-purified extracts sustain a centrifugal partition chromatography, and preparative HPLC permitted to achieve the purification protocol, leading to a structural characterization of pure molecule. And after each fractionation step, the extracts were submitted to sensory analysis to select the most active one. So there is in particular a fraction of an untoasted wood, which was interesting for its sweet taste, but it was still a little bit complex from a chemical point of view. So we decided to continue the purification protocol, and the CPC was used. After this experiment, 15 fractions were obtained, including C fraction interesting for the sweet taste. We decided to focus only on fraction 10, which exhibited an intense sweet taste, and a simplified chromatographic profile with only 4 peaks. So the compounds were named A, B, C, D, and were purified by preparative HPLC. The most abundant molecules, A and B, were already non-compound. Compound A was the lyorizinol, and compound B was his whiskey lactone precursor. So these 2 compounds were already identified in oak wood, but their tastes were unknown. After that, the tasting reveals that lyorizinol was bitter, and the whiskey lactone precursor was tasteless. So at this point, A and B could not explain the sweetness perceived for fraction 10. So we decided to continue the purification protocol, and we looked at the other 2 compounds, C and D. Tasting shows that these 2 compounds were sweet, and that they reduce the bitterness of the other oak components. C and D are isomers with an empirical formula of C33H62O15, And in order to identify these two molecules, two powerful techniques were used, high-resolution mass spectrometry and nuclear magnetic resonance. Analysis have made it possible to identify these two compounds, which are galoylated and glucosylated triterpenes, which differ by the position of the galate. These compounds have never been identified in the literature before, and we decided to name them quercotriterpenosides, QTT, QTT1 and QTT2, respectively, for C and D. So this development of this protocol has contributed to the identification of a new family of sweet molecules in oakwood. These interesting results led us to search for QTT isomers and functionalized triterpenes, and natural biosynthetic pathways generally induce structural diversity among the secondary metabolites. So for a given matrix, isomers or derivatives can exist in the same plant, and we hope that they can develop similar or more taste properties. So oakwood extract was screened by LC-HRMS, and CPC purification of targeted compound guided by LC-HRMS was performed. This protocol allowed us to purify 39 triterpenes, including 23 new molecules. And after that, all the molecules were tasted. And here you can see in the table different sweetness depending on the molecule. And they are likely to contribute to the sweet and bitter taste of oak wines. So the success of this approach that I just presented to you is based on the choice of the raw extract, the reliability of the sensory test, and of course the access to various technical analytical techniques. However, masking phenomena between compounds of the same fraction can occur during tasting and affect the efficiency of this approach. Consequently, a complementary strategy has been implemented by coupling the sensory analysis to the measurement of the activation of human bitter taste receptors. So after each fractionation step, the extracts were tested for their ability to functionally express TAS2Rs using in vitro 25 bitter taste receptors. So this original approach were carried out in collaboration with Dr. Loïc Briand and Dr. Christine Berroir from the Center for Taste and Feeding Behavior. So for non-experts and very schematically, cells expressing the bitter receptors were loaded with the fluorescent probe, which allows to measure intracellular calcium release. So when the fraction or the molecules binds to the bitter taste receptor, it will induce a calcium flux, which will be followed by fluorescence. We decided to start this new approach with an oakwood extract, a non-toasted oakwood extract, and we screened different bitter taste receptors in order to see which responded better with an oakwood extract. And the results showed us that the cells expressing 5 of the 25 TAS2Rs responded to challenge with an oakwood extract. But like I said before, we decided to focus only on the most robust response achieved with the TAS2R7 receptor. So like I said before, to begin the purification protocol, successive liquid-liquid extraction have been conducted and 4 extracts were obtained. So increasing concentration of this extract were added to the cells expressing the bitter taste receptors, and we can see that we have a better dose response from the aqueous phase and for the butanol extract. I will not show you all the fraction, but after successive fractionation, a pure compound has been purified, and before proceeding to any structural characterization, we wanted to see if this compound could activate the TAS2R7 receptor. So increasing concentration of this compound were added to the cells, and results showed us a robust dose response of this compound at 8 micrograms per milliliter, or approximately 15 micromolar. And for example, and by comparison, magnesium sulfate, which is an agonist of TAS2R7, human TAS2R7, is on the order of 15 millimolar. So we were very happy about these results. And after we carried out the structural characterization, I cannot show you the structure right now because the results will be published soon, hopefully. But I can tell you that it's a compound that came from the galloyl glucopyranosyl family, and we decided to name it GGDNC. We decided after that to taste this compound, and lucky for us, it was bitter. So this development, the development of this new approach, allow us to isolate and identify a new bitter compounds from oakwood. So these results demonstrate the relevance and the effectiveness of combining separative techniques, sensory analysis, and receptor activation to purify new compounds. So after these three main approaches, the last approach that I'd like to present to you today has been applied in particular to spirits and concerns the development of untargeted LC-HRMS profiling followed by targeted fractionation to discover new taste-active compounds. Spirits seem particularly interesting to study because their non-volatile component is exclusively conferred by the aging in barrels. So an untargeted analysis by HRMS was performed on a series of eau-de-vie from 10 vintages from 1970 to 2015. And these samples were not commercial spirit, but spirits still aging in barrels. They came from the same distillery and they age in the same condition. And there were 5 replicates per vintage. After that, we perform LC-MSMS analysis in collaboration with the Dr. Eric Ferro from Firmenich. After this analysis, the data were processed according to the metabolomic strategies implemented for natural products. And we use the EnzymeMine 2 software to do that. Statistical analyses were carried out with Dr. Warren Albertin from the Oenology Research Unit. And we use statistical analysis to assess the overall structure of the metabolomic data and to select compounds of interest. Following treatments on EnzymeMine, 42,000 ions were observed for the negative ionization mode, almost 4,000 ions in 1970 against 30,000 ions for the 2015 spirits. And you can see clearly these differences in the 3D chromatograms for each spirit. We see more chromatographic peaks in 1970. There could be 2 hypotheses for explaining this observation. Either the compounds were continuously released from wood to spirits during aging, or the compounds were neoformed by chemical reaction in the spirits. So these 2 hypotheses, we are trying to select one compound for each. According to several criteria. First, we create statistical groups and we classify the molecules according to the spirits. And after that, we did LC-HRMS screening of these compounds in spirits and in oakwood extract and we select candidates according to 3 criteria. So, for example, the high abundance of the targeted compounds, high increase in older vintages, and for the compounds that could be neoform in spirits, a large gap in intensity between the spirits and output extract. So we found 2 compounds, TC1 and TC2, with nominal mass of 361 and 703. So TC1 were purified according to the same purification protocol presented earlier and came from oak wood, and TC2 were purified from spirits, but they were evaporated to have a dry extract, and after we have the same purification protocol. So for TC1, NMR was carried out in collaboration with Professor Pierre Fotego from the Oenology Research Unit. I will call it ALK-D-HDDC because, as you can see, the name is a little bit complicated to say in English. This compound has already been identified in several species, but this is the first identification in Quercus genus and spirits. For TC2, NMR was made in collaboration with the Professor Stéphane Kido from the Institute of Molecular Sciences, and we discover a new elagitanin called brandy tannin A. After that, we wonder what could be the formation mechanism of brandy tannin A. We look into the literature and we found this compound, whiskey tannin A, which is the castellation oxidation products, and they come from a pure malt whiskey. So this compound is an analog of our compound, but we saw that with the NMR data that our compound is not a derivative from castellagine, but rather from vescalagine. And the other main difference between these 2 compounds is the presence of a netoxic group at the same C1 center. We decided to test this compound, of course, to see if it's interesting, and it was perceived as sweet at 2 mg per liter. And the same result was obtained for TC1. This compound is sweet as well. After development and validation of an LC-HRMR quantification method, these 2 compounds were assayed in different spirits, but also in the same set of spirits that we use for the metabolomic approach. And we can see that long aging seems to promote the extraction of ALK-D-HDDC. And brandy tannin A seems to follow a bell-shaped curve, lower in the young vintages, maximal in the middle, and lower in the older vintages. Moreover, 5 and 6 of the 10 vintages, respectively for TC1 and TC2, were greater than 2 mg per liter, which means that sensory studies showed us significant taste modification at this concentration, which demonstrates their contribution to the taste balance of these spirits. So in summary, the objective of our laboratory is to propose effective methodology to discover new taste-active compounds, and the results demonstrate the importance of conducted complementary strategies, but also to combine analytical chemistry and sensory analysis to discover sweet and bitter compounds. And thanks to powerful analytical techniques, we will be able to find a new elagitanin called brandy tannin A. So this study provides promising perspectives for a better understanding of the molecular markers responsible for the taste of foods and beverages, such as wine and spirits. Thank you for your attention.
Thank you very much, Dr. Winstel, for this very interesting presentation, which complements very well the presentation of Jean-Luc Wolfender. And actually, it is really amazing how the non-targeted profiling of your spirits allowed you to identify the most interesting compounds that were formed during aging or increased a lot during the aging. So really, it was an interesting presentation on a special type of bioactivity because it's a sensory perception. So now the floor is open to questions, and we have already. So please feel free to add your questions on the chat. If not done already. But we have already a first question for Professor Wolfender. So some of our colleagues is using 2D hyphenated HPTLC. And they transfer the bioactive compounds from the bioautogram online to HPLC HRMS. So the question is, can your annotation also be used in this case?
So thank you for the question. Indeed, so bioautography is a very effective way to localize bioactivity in a profile. Yes, you can use the same method somehow, because when you transfer a spot from bioautography into LCMS, if the LCMS that you can link to bioautography is an LC-MSMS method, as I presented, so which allows you to obtain not only the MS, but also the MS-MS spectra, then the data generated can be compared to other fraction or other extracts, and depending on the bioactivity you have with bioautography, you can make similar type of of correlation. And it's even better there, because since you just scrap bioactive zones, you will have only a few compounds, and you are more into the fraction. But yeah, this is normally possible.
Okay, thank you, Jean-Luc. So another question for you. Somebody working on vegetables with a high fiber content, would you recommend longer extraction time, solid liquid, to get higher total polyphenol content?
So there, I'm not really a specialist on the polyphenol aspect itself. Of course, I guess that if you go for longer extraction time, you will get better from fibers. But maybe I'm not the expert to help you on these aspects. I don't know. Maybe Delphine, you know better on this side.
No, I'm not really sure. Yeah, maybe it's different solvents, but.
Yeah, you always may have to work with different solvents and compare profiles and yields. This is normally the best way to do.
It can be also a case by case, depending of the matrix you are using. It can be a little bit different. Yes. I have a question now for Delphine. Congratulations for your presentation. Are the metabolomics techniques looking at wine also showing up pesticide contamination, such as neonicotinoid?
Good question. I cannot answer for the pesticide contamination because we are not working on this part of the project. I know there are some subjects at this moment in the ISVV that working on the pesticide, but not maybe on the metabolomic part. But in the same team, Marie Lescamp, which in PhD student with Axel, is working also on metabolomic with the metabolomic approach with red wines currently. So. But to find also new taste active compounds, I don't have the answer for the technique.
Okay, thank you very much, Delphine. So as I announced at the beginning of the seminar, of the webinar, we are almost at the end, so I will conclude now. But there are still questions, and as I offered to you, we can stay online to answer those questions. So of course, I would like to warmly thank our two presenters, Professor Jean-Luc Wolfender and Dr. Delphine Winstel, and of course, warmly thank also Professor Aude Vatrelot and Professor Juha-Pekka Salminen, who are really deeply involved in the organization of those seminars. So actually, as you know, the seminars have been recorded, and the recording will be available on the Groupe Polyphénol website, the YouTube channel of the Groupe Polyphénol, and the Instagram and Twitter accounts of the Groupe Polyphénol. So you will receive some news concerning this. And also, all the participants will receive an email concerning a survey to get your feedback on the webinar. So the next webinar is foreseen for February 2023. The topic will be polyphenols and food and beverage quality, and it will be chaired by Professor Aude Vatrelot. And after this event, we'll have this summer even a bigger event, which will be released, the International Conference on Polyphenols 2023, which will be held in Nantes from the 3 to 6 of July. So, of course, I strongly encourage you to save the date and to already register to this conference. You probably all received already information about that. If not, please contact us. So having said that, thanks to you all for attending this very interesting session. So I now close officially the webinar, and we stay online for the additional questions.